I'm here at the stand for the Wellcome Centre for Integrative Parasitology. Hi, lovely to see you.
Hi, so nice to be here and thanks so much for interviewing me.
So please tell me, what is going on here today?
So today we have the Wellcome Centre for Integrative Parasitology from Glasgow down with 10 of our researchers, all parasitologists, engaging and teaching about the neglected tropical diseases that we study.
Great, so we've got, I can see lots of different things going on in the stand. So why don't we start with this here? What's going on here?
Awesome. So we work closely with the 3D printing unit at Glasgow University, and they're working with us to create different ways to engage about the diseases. We try to use different methodologies, so we're trying to we have checkers, we're working on a 3D printed chess set where all of the pieces are parasites. We have an amazing artist that was an intern at their group that printed us all of these fantastic playing cards about the life cycles of our diseases. So that's just kind of a small way that we try to engage outside of the normal bounds of just kind of science communication.
I see. So what are the main diseases that you're talking about here today?
So we study African sleeping sickness, which is caused by the parasite trypanosoma. We study schistosomiasis, we study toxoplasmosis, leishmaniasis, and one that most people know, malaria, which is caused by the parasite Plasmodium falciparum.
Great, amazing. So behind me I can see some soft toys and some games. It looks like people are already really having fun with the games and getting involved. So should we take a look?
Absolutely, let's go.
Okay.
Hi, I'm Grace.
Nice to meet you. Can you tell me what's going on here today?
We have a fun game for everybody. It's called Host, and it's basically pairs or snap. And we've got 5 parasites, we've got 5 diseases caused by the parasites, and then we've got 5 things that pass on parasites, so they're called vectors. And we're just trying to get people to match it up and use it as an opportunity to tell people about all these cool parasites that we work on.
And tell me about the soft toys I can see over there as well. What are they?
So everybody loves a soft toy. So we've got a couple of fun ones. We've got a red blood cell here. We've got a leishmania parasite here. So again, it's just, I guess we're using it as a tool for people to be seeing things that you maybe wouldn't see unless you had a microscope. And you can play with them. And again, it's just an opportunity for us to be like, this is a parasite and this is what it does. This is what the disease it causes. It's just
They already look really popular.
Yes, definitely. I think I'm going to try and take some of them home. I think.
Good idea.
Home, yeah. So.
Great. Thank you so much. I'm going to see what else is here. So what else have we got going on?
I think we should go over to the microscopes.
Sounds good.
All right.
So what can we see on the microscopes today?
Could I borrow you to talk about what we're showing under the microscope?
Absolutely, yes. Of course. What you're seeing is 4 different microscopes. We have the larval and adult stages of a mouse parasite that's found within the guts of mice, the intestines. And you have the larval and adult stages here. And further down, you have toxoplasma, which is a human infected parasite. And then at the very end, we have, under the microscope, a malarial infective fly.
So tell me about the parasites. Are they sort of like, the ones in mice, are they like worms?
Yes, so what you see here in the H. polygyrus is it's a helminth that infects worms. That infects mice only. It's a parasitic worm. And what happens is you either drink infected water that have the larvae that have been secreted out of a mouse, and then they encyst within the gut walls, and then they burrow out, and then they coil around the intestines, and then eat all the food and nutrients that come down the way. And then they secrete their own eggs, and then pass on the infection further along. And it's a mouse models, because it's much better to study mice than it is humans and infect more humans. And what that can do is that not only can it help us provide information on how to get rid of parasites that might be infective and maybe developing anti-helminthic resistance, but it can also help us figure out maybe new proteins and new ways to develop ways to fight autoimmune disease or infectious, like other diseases that parasites might provide answers to. Much like, aspirin was found through like the bark of trees and things like that, parasites can also provide information via their proteins and secreted products that might provide insights into how to modulate your immune system to make maybe anti-inflammatory effects.
Okay, so it's not all bad.
Not all bad. So helminths, yes, they provide a lot of ways to study that, we don't want parasites. There's a reason why we started to get rid of them. If you look down the microscopes, hopefully later, you'll see why we got rid of them. But there's a balance, there's a play. Like up until a few years ago, we all kind of had parasites until we started filtering our water and things like that. So, what does that mean to have lost this thing that we evolved with? We've evolved with parasites. Yeah.
And toxoplasmosis, I've heard of that. Is that from cats?
It is, yes. It is from cats. So Mariana there is an expert in toxoplasma, and you can see the toxo underneath. Toxo is very different, so it's still a parasite. It still infects people similar to parasitic worm, except this case, you're right, it comes through cats as well. And it's the same way, in infected, you just get an infection via the water, maybe like a scratch from something that has it on the claws. And then, a human can get infected, and it can cause some really serious issues. But parasites are really interesting because they're such a huge family that it's ranging from these big parasitic worms. I mean, you think of tapeworms, they're meters and meters long. Toxoplasma is a single cell. So then it's still a parasite. It's still technically a part of that same parasitic branch of the thing. They're really scary creatures.
I know, but people seem to be loving looking through the microscopes at them today.
Yeah, I think so. I think people don't realize what's, this hidden world that's just a little bit too small for people to see, and just seeing it, figuring out where these things are. And in the UK, Europe, and America, we've kind of forgotten that parasites are around. That's why we've called it neglected tropical diseases, because for the most part, except for malaria, they're pretty much all neglected. And people forget that these are just around the corner. I mean, certainly your livestock in the UK haven't forgotten that it's still infecting a lot of cows and sheep and things like that. But yeah, I think people are really trying to remember what it was like to have these as an everyday occurrence. Yeah.
Amazing, it's fascinating. Can't wait to look through the microscopes. Thank you so much.
Please do, and come down. Yeah. Thank you.
Thank you. Lovely to meet you. I'm Abby. Can you tell me a bit about what you're doing here today?
Yeah, of course. I am trying to teach people what leishmaniasis is and how important this disease is worldwide. Because a lot of people hear about malaria and how important it is, but they don't know that leishmaniasis is actually a parasitic disease that kills the most after malaria. And it kills and affects the poorest of the poorest populations worldwide. So we are trying to tackle that and at least try to teach people what leishmaniasis is and what we do in the Wellcome Centre for Integrative Parasitology.
And what can people do today at your stand?
Well, they can definitely touch a lot of things. They can touch our 3D printed prototype of a sandfly, which is still ongoing. And they can learn that sandflies are very different from mosquitoes. They can also learn a little bit about how the parasite develops inside the sandfly. So I'm trying to teach them how a sandfly takes up a blood meal and how that blood meal looks like and how the parasites that survive inside the sandfly actually get into the mouth parts of the sandfly and then infect again the next host, how the parasites then survive inside the host, and how we use this information about the biology of the parasite to perhaps find novel drugs to treat.
Amazing. So are these the parasites just here?
Yeah, that was a very boring weekend that I had without internet, where I played a little bit with Fimo-clay and made a bunch of different parasites. So some of the features that you can see here are the tail, which we call a flagellum. And I try to teach that this tail is very important to attach to the gut of the sandfly and also to move inside there. And I am also trying to explain here with this model how parasites and our cells are so similar, and that's one of the reasons why the drugs that we have available to treat the disease are so toxic for us, because they just affect the same types of components in our body.
Yeah, okay, so are these our cells here?
Yeah, this is an animal cell here, and you can see all of the organelles. So you have basically the organs of the cell. You have a nucleus with DNA, you have the Golgi apparatus, the endoplasmic reticulum, and the mitochondria. And if you look at a parasite, you also have some of those components. So I'm just going to put this here. You also have a nucleus there in orange. You have a Golgi apparatus. You have a big mitochondria. That's one of the things that is different between us and these parasites, which is have one mitochondria in the parasite and a bunch of different organelles. But basically, when these drugs are used to treat this disease, they are going to affect very similar components, and that's what makes them so toxic. So we try to use, for example, in my work, I try to find the Achilles heel of these parasites by genetically modifying one gene one by one, try to find which one actually kills the parasite, so that we can then sort of liaise with drug development companies to just target that one thing that will kill my parasite.
Amazing. Thank you so much.
Thank you for coming.
We've got more going on over here. Hi.
We research in our centre is Schistosomia mansoni. So it's a worm which infects people in Uganda, for example. And the way you get it is by swimming or being exposed to an infected water body, so a lake, which is often a problem because life is all centered around water. So we wash our clothes in water, we get water, it's hot, we swim in the water, and we get infected by the parasite which invades our skin. Once we get infected, we have it in our liver, where a lot of eggs are produced. We go to the toilet, and that's how the cycle of life cycle is completed. If we're unlucky, the sanitation is not that good, a rainfall comes and it gets flushed back into the lake. So what we do in the field is we try to test and treat. So we have parental control available and we can treat. And the easiest way to test in the field is using a rapid test. We know this test probably ran out from COVID, and I can show you how to do that.
Oh, great.
So we've got urine here.
Is this really urine?
I'm not going to tell us. It's from my friend, but I don't know. So we've got a test. And so it looks like a COVID test.
It looks exactly like a COVID test,
so it's like a rapid test. So the principle is, again, we have a control line, so that will go off if the test works, and a test line that will go off if the person is positive. So we've got a bit of urine in this jar. So we have a Pasteur pipette. We take a few drops and we put them on our little test. And we wait.
How long does it take?
It takes a couple of minutes usually, and what we test here is an antigen called CCA. So CCA stands for circulating Catholic antigen, and it's actually released by the eggs and also from the worm. So if a person is infected, you will have a large amount of this CCA potentially in your urine. So we can see whether the person is infected.
Great, thank you.
Whether my friend, who I'm not disclosing who it is infected, we'll see.
Okay, yeah, let's have a look.
So yeah, you can see it will travel up and it will hit the test line. And then once it reaches the control line, it will give you a green line.
So this is something obviously most people will be familiar with COVID testing, but has this been around a lot longer than that?
This has been around a lot longer. There are actually two different types of Schistosomia mansoni. So this one is we can find in the urine. The one which is more prevalent in Uganda, it actually gets transmitted in the stool, so in the poo. That makes it way more difficult. Poo is a bit more it's hard. You can't really do a test like that easy. So you would have to do microscopy, smear it, and look for the eggs. But there's research going into improving.
Amazing. Thank you. Thanks so much for showing us. Hi, nice to meet you.
Hello.
I'm Abby. So can you tell me a little bit about what's happening today?
So today we are here with the Wellcome Centre for Cell Biology doing science tattoos. So we have a project called Tattoo by Science, or hashtag Tattoo by Science for social media, and we are basically taking research images from our centre and turning them into fun tattoos for people. We've got an option for either transfer tattoos, which is mostly for the kids, but the adults love them too, and then also henna tattoos. And these are, as I say, all based on designs that our researchers are actually working on, and it's our researchers that are delivering, so that in that time that it takes to deliver the tattoo, we get real quality dialogue going on between our researchers and the public.
Amazing. And so it looks like it's already really popular. What are the most popular tattoos that people are getting?
So for our hennas, things like the DNA helixes, because obviously that's a really familiar concept to people, they'll choose those. Another very popular henna one is our base pairing. So the idea is that with your best friend or your partner, you can get a matching base pair and they line up on their wrists. In terms of our colourful transfer tattoos, our jellyfish always is the big hitter. In fact, we've had to cut up about 100 more jellyfish today because it was so popular with the schools yesterday.
Amazing. Thank you so much.
Very welcome.
Perfect.
We have some of our amazing science comics. We co-developed them with an artist in Edinburgh, Eddie Ross, and they started out about 15 years ago with one of our researchers, Jamie Hall. And we now have collected all of the parasites we're working with at the center, except Leishmaniasis, however, which is this beautiful squiggly parasite right in front of the world. But our Leishmania comic is coming up soon, and our comics feature researchers from our center. So you can see on the front, this is Dr. Poppy Lamberton, who's actually speaking today, and this is Professor Rick Maisels, Professor Poppy Lamberton recently. Congratulations, Poppy. And so these are some of our researchers that we feature in the comics, and we also have parasite postcards, so if you want to come by the stand and pick one up, you can mail it to us and we'll put it on our parasite postcard wall. And if you want to download any of these comics, we are trying to get them into every language possible, so they're more useful in our endemic regions. They're available for download on our website, or you can tweet us at WCIP Glasgow, and we can send some out to wherever you are.
Thank you so much.
Of course. Thank you so much. This has been such a pleasure.